Hey, what's up, everybody, and welcome back to the Sanctuary. I'm your host, Professor C, and we're going to be doing a little bit more A&P. Specifically, we're going to do the third part of the Central Dogma, which is translation of protein, and understand a little bit more about the genetic code. So, this is pretty cool. Let's get to it. Remember that mRNA is a copy of a gene. It is the recipe that needs to be cooked up by the ribosome. I like to use these cooking analogies. Ribosomes read it, but they don't read it as you would read, say, a piece of paper. They're going to read it 3 bases at a time. And these bases are called codons. Codons, like the unit of a code, right? We're going to try to talk about the genetic code in a moment. And what we see during translation is kind of a decoding of this genetic code. So the unit of the code is a codon. The codon is a triplet of bases, 1, 2, 3. Each codon represents one specific anticodon, which will translate for one specific, only one amino acid. 1 codon equals 3 bases equals 1 amino acid. All right, so 1 codon is 1 amino acid. A large subunit is going to be the main subunit of the ribosome that's going to do the business. Remember, we talked about having a big subunit and a smaller subunit, kind of like hamburger buns. We're going to see some special places inside of this big ribosome that kind of make it look like Daffy Duck or something like that to me. Sometimes when I see that, I see a duck face. And there's going to be facilitation of bond formation. We're going to create the peptide bonds inside of the ribosome, which I mentioned is kind of like the ovens of the system that's going to cook up our protein. So in order to deal with this topic, we need to deal with what's called the genetic code, and we need to be able to read it. Now, I've never had a class or anything that made me memorize the genetic code, so just being familiar with the chart is good enough. But the point is this: a codon is 3 bases, X, Y, and Z. And if we're talking about RNA, this first base can only be one of 4 things. It could be a G, an A U, or a C, right? Because we're talking RNA bases, 1 of 4. This middle part of the codon, the middle base in the codon, it could be a G, an A U, or a C. That's our only options. So that means this third codon, in this third position, it can only be a G, an A U, or a C. All right, so what I'm doing is, if the first position could be one of 4 things, and the second position can be one of 4, and the third position can be one of 4, we can do a little math, and we can do 4 by 4 by 4, and probability tells us there are 64 different ways of recombining 4 different things 3 times. This says there are 61 codons. And you may be confused there. We just went over the fact that there's 64. So what I'll say is finish the sentence, all right? There are 61 codons that code. That must mean, since there's 64 total possibilities, that 3 don't code. All right? So yes, there are 61 codons that code for an amino acid, and there are only 20 amino acids. 3 of those codons of the 64 do not code for anything. They're going to be a stop codon. The redundancy, and what I mean by redundancy is, if there are 61, let's just say 60 for the fun of it, and 20 amino acids, it looks like there's a 3-to-1 repetition. Right? If there's 61 codons but only 20 different amino acids, that's about 3-to-1 at least. And you can see that if you look over at the genetic code here on the left side of your screen, you can see how CGU, CGC, CGA, CGG all translate to amino acid arginine or ARG. We're not going to learn all the names of the amino acids. This redundancy in the code is called degeneracy. So we do have a degenerate code. It is redundant. Not only a redundant, but triple redundant, right? It's got a 3-to-1 redundancy. The reason for this is that it protects us against mutations of some types. And to get into those details, I'm saving for further down the road. There is one specific codon that I'm putting a box around there that is very important. It's called the start codon, AUG. And if you just remember that translation begins in August, then you'll never forget that one. That's pretty easy to remember. It will be start, and not only is it the start of your cells, it's the start for every life form on the Earth which uses this code, which is all of them. So all life forms on Earth use the same start codon. We call it a universal start codon, AUG. It tells the ribosome where to begin adding amino acids, right? It tells the ribosome where to begin translation. The first one will be MET, and that's what this MET is. If you look over at the code, I'm underlining MET here. MET stands for methionine. Again, you can learn the long versions if you want, but the codon AUG translates to amino acid MET. Remember, each triplet gives us one amino acid. So what that says is, because AUG is a universal start, every protein, not just made by you, but every protein made by every organism, which is all of them, begin with amino acid MET. There are 3 codons. Remember we said 3 don't code. And you can just go right here. The 3 that don't code are the 3 stops. UAA, UAG, and UGA. Here's the way that I used to remember it. It's very silly, but dang it, it works. I used to watch football back in the day while I studied for class, because there were a lot of commercial breaks back then, and I could watch the game for a little bit and then take a break and during the commercials and study. And so here's the deal. There's a game today, University of Auburn at Georgia, or is it University of Georgia at Auburn? Can't remember. Either way, ooh, ah, ah, let's go watch them play football. Right? It's super silly, but it works. Let's try it again. The University of Auburn at Georgia, or is it the University of Georgia at Auburn? I don't remember. Either way, ooh, ah, ah, that's going to be a great game. Let's go watch some football. So that's the 4 that I would remember off of anything on this code over here. I would remember the 1 start, AUG, translation begins in August, and the 3 stops, UAG, UGA, and UAA. Otherwise, just refer to the code as necessary in order to translate. And we're going to pretend to ribosomes in a minute and try some translating. All right, so let's check it out. This little ribosome here, and that's what that's supposed to be. This is supposed to represent a ribosome. You see the big subunit on top. You see the small subunit on bottom. And this right here, this little green strip of stuff is supposed to be mRNA. So that's what we're picturing. We're picturing the 2 buns, if you will, of a ribosome. Remember, you have this kind of big bun sitting on top of the smaller bun. And then in between the 2 buns, sliding in between, is this mRNA. And that's how it's usually drawn on many figures, is just like that. That's what I'm trying to draw in here. Now notice how inside of the ribosome there are some special sites we've marked as being a P site. Here's the P site, and here's the A site. Some books may mention an E site that would be to the left of P. I don't necessarily need to talk about that site in order to deal with the process of translation, so I'm going to ignore the E site, even though I am aware that it exists. Okay, so translation begins when the ribosomes bind to some mRNA, just like I've shown in the picture, and we find a start codon. Again, critical, we find the start. What about anything in front of the start? Well, it's just like you're going to work and you're supposed to start at 8 o'clock. Well, you're not going to start at 7:55, right? So this here, if you're a ribosome, you don't start early. You start when it says start, and you will ignore anything that's upstream from the start. And we know what's going to happen. And tRNA is going to fit right in here. I'm going to draw it just like this. And remember we had these 3 whiskers down here? That's going to be what we call an anticodon. If the codon is AUG, then this would bear the anticodon, right? Not a T, but a U, A, C. Now that's unimportant if we're trying to read the code. We just want to read what's on the mRNA. but it would bear the anticodon UAC. And there's only one thing that a tRNA with the anticodon UAC has hanging off of it, and it is the amino acid MET, methionine. And we knew that already. Translation begins in August, and the first amino acid is MET. And it says right here, a tRNA with the appropriate anticodon. Let me go ahead and circle that for you a little bit better. With the appropriate anticodon, in this instance UAC, carries MET and sits in the P site. Now this is the only one of all the tRNAs. This is the only one that goes into the P site first. Right? Everybody else is going to have to go into A and then shift over to P at some point in the future. But being the special first guy, he gets to sit in the P parking spot immediately. Right? And he's carrying MET. Now, we only have room inside of this ribosome for 2 tRNAs, right? Excluding the E site, which we're skipping completely. So I do have room. And let's go ahead and draw another tRNA in here. Beautiful. There's his anticodon. What would it be? Well, a U would hook up with an A C would hook up with a G, and a C would hook up with a G. So there's our second tRNA. It will go in the A site, right? Because the P is already filled by the first guy. The question is, what is he carrying? Right? And that's what it says here as the next statement. The adjacent A site is occupied by the next matching tRNA. But again, which amino acid is he carrying? Well, I know the codon is UCC. And if I flip back real quick and look at my genetic code, I look around, I can kind of pan and scan for UCC. Anybody see it? There it is. UCC, and this codes for the amino acid SER, or serine, if you want to get fancy. So we could go back to our picture here, and we could put SER. And this is how we get our first 2 amino acids in place. We can't put any more yet because the ribosome is full at the moment. There are only 2 spots to sit. So what must we do now? Well, probably what we want to do is combine these 2 together. And that's what's going to happen in that catalytic site. We're going to get a peptide bond. Remember, that's the bond that holds amino acids together. A peptide bond will be formed at that cat spot, what I'm calling the catalytic site. So we can't just stop there. 2 amino acids do not make a protein, not even a polypeptide. We need to add potentially hundreds more in this chain. So let's see what happens next. Another thing you can do is think of these tRNAs that are in these P and A sites as kids, little kids, children, holding balloons. And at some point, their balloons get tied together at that catalytic spot. And what we're going to see next is the first kid is going to just let go of his balloon and let the second kid have 2 balloons. So let's check that out. Okay, after forming the peptide bond, the ribosome releases the first tRNA. So let's show that happening. Okay, there's the first guy. He's on the way out. So let's kind of show him getting out. Now remember, in some books, you'll see that he'll enter an E site, an ejection site, and he'll get ejected. But we're just going to kick him out right now like James Bond. Just get out of here. He's ejected. Now remember, he doesn't have a tRNA. I mean, that tRNA doesn't have an amino acid on him because he's already given it up. Right? He's already given it up. So he's going to head out. Now this guy who is left here, he had a SER on him, right? But now he also is carrying the first kid's balloon too. Now this guy is going to come over here. Whatever this next, remember, we're going to slide through the buns here. Whichever codon comes up next, we'll get the next tRNA to come out from here somewhere. And let me just draw them in yellow so you can visualize this about to happen. And he's going to have his own amino acid on here. Who will it be? Well, look at the code. It says GCU. And if I flip all the way back to here and I find GCU, it's right here. If you can see my yellow pen, alanine is the amino acid. So let's go back and label that ala. It's almost labeled for us. I'll just cheat and put an L here. There's alanine is next. And we'll have the same game over and over and over. And now we can move on to the next step. As you slide along and you keep doing this, you get a new tRNA. And let's just draw the picture as it would look right here. This guy could be saying eject, and you're gone now. Get out of here. The next guy's coming over here. The next guy's going to come in here. And eventually, you know, this guy, oh, he should only have 3 whiskers there, not 4. What's he going to be carrying now, right? We looked up GCU. It was ALA, right? So he's going to be carrying an ALA, a SIR, and a MET. Right? And then we're just going to keep on playing the game. The next guy, A goes to P, a new one comes into A. We bond at the catalytic site, and we kick him out, and we shift over, and then we kick out, and we shift over, and just repeat, repeat, repeat. How far do you go? Well, here's the first question. What happens if you come across another AUG? This often happens, and it's a trick on some tests. They'll put another AUG as another codon. The answer is you don't start over. You just put another MET and keep on chugging. Remember, we only stop until you get to one of the stop codons, UAG, UGA, or UAA. So if you encounter another start or 2 more or whatever, you don't restart. You just chunk in another MET, methionine, and you keep on going. That's what it should say here. We're going to keep on chugging and chugging and chugging. And if you want, you can draw these little fellas in here. But what's really important to me is that I see a stop coming. So at some point, and I'll just kind of exaggerate it here so you get the point, this guy will be here. Some guy will be told to get the heck out of here. And then this dude is going to be carrying a whole bunch of balloons, right? So many balloons that I got to change the color because they get off screen here. More and more amino acids, and you could follow it all the way back up to the first one, which will be MET. But he's got a whole bunch of balloons now. But now I see the stop codon coming, UAA. Remember, there are 3. Let's just write them out again. UAG, University of Auburn at Georgia. University of Georgia at Auburn. Ooh, ah, ah. Watch them play football. There's the ooh, ah, ah part. And what happens at the stop? Everybody stops. Once we stop, the game is over. The ribosome floats away. The mRNA floats away. And kind of looks like this picture I'm about to show you here. Once you hit the stop, no more amino acids are incorporated. Frankly, because there are no tRNAs that have a matching anticodon to any of the 3 stop codons. That's why nothing is put in there. So the ribosomal parts break off. The mRNA floats off somewhere, usually digested. And then the polypeptide, right? It's not quite folded up yet, so we'd call it a polypeptide at this point. Floats off too. Later, hopefully, it will be folded up into a functional protein. And we did a talk a long, long time ago in Unit 2 about how to fold proteins. Okay, wonderful. There's the process of translation. Let's play a game. Let's pretend we are a ribosome. Let's actually do a translation event if you want to practice it, you know, for real, right? So if we want to know we're doing it correctly, there are some questions we could try to answer to make sure we're doing it correctly. So let's start with this. How many codons are in the reading frame? Now, what the heck is a reading frame? Well, it is exactly like you would think it is. The ribosome is going to read in between a start and a stop codon, right? So that's the reading frame, is, you know, how many codons are between start and stop, including start and stop. So let's play this game up here. Now, I can take my mRNA, and I've got it clearly labeled here, this mRNA. And I can, you know, look for my start, and I can try it that way, but I see several AUGs in this mess. And so what we want to do is we want to find the first AUG. Remember, we're not just going to start breaking these into triplets and translating them because we don't know where to start yet. So what do I need to look for? First of all, what I need to look for, the start codon. I see a couple, but let's find the very, very first one. And here it is. Just to make sure, look in front of it and make sure we didn't miss any starts. And we didn't. We know translation begins in August. What does the ribosome do with these bases? It ignores them completely because it's before the start. Now we can pretend that we're a ribosome, let's clamp down on this and let's do the translation without actually doing P site, A site, P site, A site over and over and over. So what I would do next after I find the start, I would not look for the stop immediately. I would break this now into codons. So let's do that. Be careful not to get a fourth one in there, or be careful not to put just two. Now what I'm doing as I'm doing this is every time I'm making a bar, I'm seeing if one of them, is a stop codon. And that is one, right? There was not one in front of that. UAG, UGA, there's UAA. That's the first one I've come across. So what do we do with the bases after the stop? Ignore them completely. We don't need those anymore. The ribosome doesn't care. What I could do is put a red box around the stop, now that I know where it is. And now I have a reading frame. I could put some boundaries on this, maybe in a different color so you can see it. Here and here, anywhere in between there is called a reading frame, including the start and the stop, because the ribosome will read the start and the stop. So it's asking you how many codons are in the reading frame. Well, I know that UAA does not put down an amino acid, but it is a codon, right? So I have to count it as a codon, and just let's count out loud. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. There are 13 codons in the reading frame. Yes, you count the stop there, even though it doesn't code. We'll deal with that in the next problem. How many amino acids will there be in this polypeptide we're about to build? Well, remember again, stop means stop. Now start, that'll put in a met. We're fine with that, and so will all the other codons. They will put in their own amino acids. So how many amino acids should be here? One less, 12, because the stop doesn't code. It's still a codon that's in the reading frame, so we have 13 codons in the frame, but only 12 of them actually code for an amino acid. And then sometimes you might be asked to list those amino acids and in order. And here's where you would just use your genetic code and do that very quickly. So let me pause and do that for you. And you can try it at home if you wish. So let's pause. And by the power of magic of editing, there is the list. Confirm to see that you came up with the same ones. If I made a mistake, send me a nasty comment telling me how stupid I am. So you can count them up yourself. 12, right? There's 12 amino acids. UAA doesn't code. That's how to pretend you're a ribosome and do some translation. Pretty fun, huh? Thanks for watching. If you enjoyed this video, check out some more in the series. Lots of stuff to learn. See ya for the next one. Bye-bye.